In this video, we're going to talk about the organic compound group of lipids. Lipids are a little bit different than the other organic compound groups, and you'll notice this on your graphic organizer. There's not a space to draw monomers and polymers because lipids do not have monomers and polymers. But because they're still found and build up living tissue and found in living tissue, and they're made of carbon, they are considered organic compounds. In fact, in many of the other organic compounds, if you break off all of their functional groups, you end up with a long hydrocarbon chain, which is essentially what a lipid is. So lipids are the exact same three elements as carbohydrates. They're made up of carbon, hydrogen, and oxygen. It's a very diverse group. Fats, oils, and waxes all are found within this group. And while the notes here say that a monomer is made up of lipids called a fatty acid and the polymer is a little different, we really don't want you using this terminology. We want you to recognize that lipids are long hydrocarbon chains. So what I mean by that is just long chains of covalently bonded carbon, and all these extra bonding sites would be hydrogens. I'm not going to fill them all in, but these would all just be hydrogens. What happens then is that the hydrocarbon chains can be attached to things. Sometimes we call them backbones. This picture here is an example of a phospholipid, and this lipid will be very important when we study cell membranes. This same hydrocarbon chain that I was messily drawing here is what is being shown right down here in the phospholipid. So a phospholipid is a glycerol backbone and then two fatty acid chains coming off. A glycerol backbone and two fatty acid chains coming off. Regardless of the structure of the lipid, the important characteristic that they have to have is that they do not dissolve in water. We spent a lot of time talking about how water is polar. Lipids are the opposite. Lipids are nonpolar, and they will not dissolve in a water. Another important characteristic of lipids is that they store energy very efficiently. This would be a long-term, very efficient energy storage. In fact, they can even store energy better than carbohydrates, and you'll explore that a little bit more in your lab work. Other important functions besides energy storage are listed here for you. Lipids are what make up all your cell membranes, and they prevent desiccation, which is just a fancy word for drying out. And that makes sense because since they can create a water-type barrier, they can keep water from evaporating and prevent things from getting dried out. As I said on the last slide, there are many different types of lipids, and here's a few that you should just be aware of. The lipid that you guys will actually model in our lab in class is called a triglyceride. It's going to be different from that phospholipid we were just talking about. Now the phospholipid, here it is again, the glycerol with two hydrocarbon chains coming off of it, or two fatty acid tails. The triglyceride, you can see, three tails, has a backbone, glycerol backbone, and it has 1, 2, and then 3 tails coming off of that backbone. So we don't technically refer to these lipids as polymers. Instead, the better term to use would be macromolecule because they're large organic compounds. Triglycerides are very common in the things you eat. All the oils that you cook with, whether they're liquids or solids like butter, are triglycerides. The phospholipids, once again, are very important because they are what make up all of the cell membranes in your body. Now, in the lab, when you are modeling and when you're building these, you want to pay attention to that basic overall structure. Essentially, what you're looking at is just a short carbon chain here and then long carbon chains here. They're going to have a special attachment that we'll talk about in class and that you will want to sketch onto your graphic organizer now. Waxes are also considered to be lipids as well as steroids because of their structure. If you look, the structure here of a steroid or cholesterol is a little bit different. It looks visually different because it has the rings. That doesn't look the same as the triglyceride or the phospholipid, but because they don't dissolve in water, they still fall into this lipid category. So just remember, while the lipids are still considered to be organic compounds, they are different. We will not identify a monomer and a polymer. We'll just identify these large polymers or macromolecules of the different categories. The test for a lipid is the simplest test that we will do for organic compounds. You've probably done a test like this before if you've ever gotten some French fries in a brown paper bag from your favorite fast food restaurant. You take a few drops of the solution that you want to test and put it onto a brown paper towel. Now, it does have to be brown paper towel. Be careful because some of our classroom paper towels are white. And then wait for the solution to dry. When the solution dries, if it contained a lipid, the paper towel will become translucent and you will be able to almost see through that paper towel. When it is no lipid present, it will just completely dry and you won't see the spot. But you do need to make sure that you run this test early in the class period so that it has time to dry. And as always with all of your tests, I would really suggest running this control so you can compare. Because if you're standing there wondering if it's translucent or if it's not translucent, it'll be much easier for you if you have that control that has been run.